All right.
See that.
Thank you for coming. For those who don't know me, I'm Katie. I'm an assistant professor in MMI, and I'm interested in kind of viral genomes, and that's how I kind of came to know Dr. Sean Sierra-Patev, who works at RIDO. He's a public health bioinformatician. Your official title is Senior Environmental Lab Scientist or something, which I guess is a big remit and doesn't quite, I think, do
yes
doesn't give you too much kind of insight into exactly what it is you do. But I think as Sean's going to talk about, he is responsible for analyzing all sorts of different genomic data that comes through the state health labs from all sorts of different contexts. And I know there's a lot of people at Brown who also work in genomic data and think a lot about the challenges and opportunities presented by genomic data, and we had some of that in the thesis defense earlier this morning. So I thought the Data Matters seminar was just a really cool opportunity to connect with other people in the state who think about genomic data, but maybe in different ways. Maybe some of the challenges are the same, maybe some of them are different. So I hope we can kind of discuss more about that after the talk, but for that, I'll leave it to you, Sean.
Thank you. Yeah, thank you so much for having me. I'm super excited to talk to everybody. This talk has really one important goal, which is to let everybody know that we're doing cool stuff in the Department of Health and to come talk to us about it. We love talking to our friends. The labs are going to be moving right into the jewelry district soon. You can come down and get fish and chips at Dune Brothers and then come hang out and say hi. Cool science is happening, and we know that you guys are also doing cool science, and we really want to have these open conversations about it because we're all working on the same stuff, and maybe there's some good ideas in there. So I have my jokey little title here of Big Data in the Littlest State Public Health Lab. We might not actually be the littlest public health lab. There are some states which are a bit smaller, but not that many. I think it's a pretty close approximation. So, but if anyone from Delaware is watching or something like that, I don't want to offend them too much. But I figured I'd let you know who I am. Why have I showed up to talk to you? Though I've had a pretty good intro already. So I'm Sean Sierra-Patev. I'm the bioinformatician down at RIDO. My title is environmental laboratory scientist, which just means, oh, okay, there we go. Which just means that, do I have to unmute or am I okay? I'm good? Okay. That just means that I'm not clinical. I'm not a clinical scientist. The way that I do a lot of work on clinical related isolates, but it's for public health surveillance work. It's stepped back a little bit, and I work very hard to anonymize rather the data, remove it from the really, really rich patient metadata that we have, and then I can provide them kind of as a bundle. Other people can link those things, people who are clinical scientists, although I am kind of actually working on clinical stuff. There's a whole approval for that. Big things are happening in the RIDO lab. My PhD is in mycology. I am a fungal systematicist, fungal evolutionary biology. I like to make phylogenetic trees of fungi. That's what I did for my PhD. Believe it or not, this is going to come up like 2 more times in the talk. It's important. You wouldn't think so, but it's very important. And I've had a steady march from extremely applied science. I used to be a plant pathologist. I drove pickup trucks all over New England, poking dead tomatoes and potatoes and a whole variety of different organisms, looking for really obscure and strange pathogens. And to answer questions about those, because I do love lab work because I am slightly lazy, and I was always like, it's the future. We can use molecules for this now. Moved into some molecular diagnostics, moved into transcriptomics when I was at Cornell working on Phytophthora infestans, which if anybody has Irish heritage is probably why you're here, because it caused the late blight potato famine. And it's still doing bad stuff now. Very cool, but very difficult bugs. So big questions that really you need a lot of computational biology to start getting at. And then moving into fungi with big, unwieldy dikaryon genomes that I can talk about all you want later on, that are also really tricky and difficult, to where I am now, where I'm almost exclusively computational. So it's been a journey, but strangely, it's not an uncommon one. I have met so many people at other state health labs and in federal programs who have almost exactly the same career trajectory that I had. In fact, I met one person who we worked in the same plant pathology lab, offset by about 4 years, doing the same project. They picked up it was a disease of basil that I had been working on. They picked up the research after I left, and then we diverged again, and now we work in 2 of the same positions in public health labs. Super weird. We're all over the place. I don't know why. I have theories, but I don't necessarily have to speculate on them too much. What is the state health lab? I get this question a lot. You are forgiven for not knowing. It's in a weird place. So we're the laboratory branch of the Department of Health. We have a building that's separate from the nice at least on the outside looking Department of Health building up on the hill. We're on the other side of the railroad tracks down near that kind of not so pretty looking hotel. We do a lot of lab work down there. There's a whole bunch of us, and we do really, really good stuff. And we work very closely with the rest of the DOH up the hill, generating data or processing data that are involved in all the products all over the DOH. There's a whole lot going on. We're very busy. We have 4 primary divisions: environmental, clinical, toxicology, and forensics. Almost all of my work right now is from the clinical side of things, although I do environmental work as well. So this can be, samples from patients, samples from healthcare facilities, samples from food, right? Or food services. Someone swabs a deli counter. That comes to us for sequencing. And it's expanding all the time, moving into wastewater, moved into wastewater, but moving deeper into wastewater, surface waters. I'm trying to get a lot more environmental data collection going on for shellfish. I have a real pet obsession with foodborne illness in shellfish in Rhode Island. There's plenty of shellfish around here. If anybody has ever heard anybody with some oysters they want to sequence, let me know. We'll talk later. But it's always expanding. There's a lot of interesting things happening, and we're trying to get into really every aspect of that. And what does a bioinformatician do in a public health lab? This is a weird term. It gets thrown around a lot. There's 1,000 different explanations, and they're all different. In the context of what I do, we are writing software, deploying software, and using software that's already out there to address these biological questions. Usually the question comes first, and then we have to try to figure out how to do it afterwards. And those questions come from all over the place. For the most part, a lot of us are pretty heavy on the biological insight side of things. Most of us are molecular biologists or microbiologists who kind of wandered our way into computational bio, but not always. And that's changing very, very rapidly because the questions are scaling very, very rapidly. And we're working really closely with a lot of these people who have a lot deeper understanding than I do of these side of things to solve these problems. Standardization and infrastructure is playing catch up, like extremely. Someone joked that things move kind of slowly in academia. Well, they move a lot slower in the lab. Validation takes a long time. Changes in these practices takes a long time. So the questions and the methods are already there, and we're just finding ways to work them into the workflows that we're doing basically as fast as we can. Truly, we're filling those roles as quickly as we can come up with them, because there's just so many great questions we could be answering that we haven't been yet, and there's no reason not to, so we are. So what has the timeline of sequencing data and data analysis been like in RIDO? It's not a long timeline, from 0 to 3,279. So sequencing started at the Rhode Island Department of Health in 2016. We didn't have a genome until then, the very first genome. And it was part of this federal pilot, the PulseNet program. PulseNet for pulsed field gel electrophoresis. You chop genomes up into little bits and you run them on a gel, and it makes a weird pattern. And you can compare patterns between bugs, and it'll tell you what lineage they are or what specific subtype and subspecies they are. It's time intensive. You got to take photos. There was this really archaic computer program that we used until like 2 months ago with the gray square buttons that looked right out of Windows 95. It was amazing, unless you had to use it every day. To compare these things. And so there was this pilot in 2016 where they were like, hey, what if you did some genome sequencing too? And then instead of using these images, we can compare genomes. It started in Listeria, only in Listeria. And when I talk about bare bones pilot, I mean bare bones pilot. You gave them accession numbers out of a table in an Excel spreadsheet you had on your computer. You emailed the guys at NCBI and asked them if you could send the genome, and then you emailed the genome over. So I hope it wasn't too big, because it had to fit in an attachment. That was it. There was no databases. There was no accessioning. There was no nothing. It was just, can we do this? And we could, and we did. In 2019, the program expanded. They got databases. They still integrated them into the program that was supposed to be for this, but they were there. And we expanded the number of pathogens. Now, this was before my time, so this is secondhand info that I'm giving you. But it expanded out to mostly other foodborne pathogens. We had E. coli, salmonella, some Vibrio, things like that. But this was the very, very beginning of actually being able to apply something to this data. So you could build these really rudimentary phylogenetic trees. It's even a bit of a stretch to call them that. You could calculate allele distances based on these very small sets of alleles that were standardized for the different species. You could get a species type and a subtype. And we started picking out outbreaks, national outbreaks with this, and it expanded internationally, actually. And we started being able to pick these things out. And in 2020, the Genome Tracker project, which is my favorite federal program that I work on, launched. And that program included environmental samples. So instead of just the clinical sample of someone who ate some bad meat, you could also swab the deli counter and include that genome. which if anyone's making phylogenetic trees, that context is super, super important. But at that time, we're talking about 160 genomes a year, maybe, bacterial genomes, which is a lot. I mean, that's a lot of work to do, but 160 a year. And then something happened in 2020. I don't know if you remember. The COVID-19 pandemic happened, and suddenly sequencing was at the forefront. This really, really moved forward to what we were doing. The response was all around genome sequencing, and it picked up in earnest. So that first real year, 2021, all the way through 2023 before it slowed down, we went from 160 genomes a year to a little over 3,000 coronavirus genomes a year. And at that time, our sequencing staff was one person. We had one person in the lab who was doing all the library prep. We don't have any space for automation. We don't have big robots. This is bench work with your hands and a pipette. 3,000 genomes a year, and trying to do the data typing for this. Okay, so the in-house analysis at that point was really just tools that were included with basically with our Illumina product stuff on their cloud. That was just real quick typing and assembly. It had some problems that I've talked about that I won't grab about too much here. But as it started to pick up and we added staff, we picked up another sequencing technician, and then we picked up me in 2022 to kind of take some of that analysis load off of them and submit the metadata. It was not streamlined, believe me, trying to submit all these sequences. We shifted from this sort of purely surveillance model where it was like, we have a listeria, into the database you go. Hope something happens with that, to actually being able to start thinking about this. You know, we have all this sequence data. We were generating thousands of genomes a year. What can we do with it? Well, we started getting questions from the medical examiner, questions from our colleagues and partners in academia about some interesting data sets that we had, right? People who we knew what their vaccination status was, people that we knew had been treated with different antivirals. We had genomes for all these people. We could supply the data, we could supply really rich metadata, and we could start actually, doing stuff with it, shifting models. And so that has kind of grown to what we have right now. So as we speak this year, I don't know for 2025 because we haven't finished it yet, but for 2024, we were doing about 500 pulse net bugs. That's the foodborne outbreak disease bacteria, about 100 of those environmental genome tracker samples. We're still at like 3,000 COVIDs a year, although this year has been a bit slow, I must admit. So I don't think we'll hit that number again this year. Special projects on invasive strep, which I'm going to talk about later. We really expanded our virus portfolio into flu and RSV. And then these really targeted studies in mpox or monkeypox, different STDs, AMR surveillance, antimicrobial resistance surveillance, which really expanded with long-read sequencing on the bottom there, because we could sequence plasmids in entirety and do separate phylogenies for the plasmids, different from the bacteria, to look for a plasmid, interspecies plasmid transfer in healthcare settings, which happens. It's really cool to find, but really difficult to do if you don't have that kind of infrastructure set up behind you. So from the 0 to 3,000 a year, what sort of data are we generating? Well, a lot of raw sequence data. And I'm now realizing that maybe I don't have to get as detailed with everybody, but I'm going to talk about it anyway because I love sequencing data. We keep all the raw sequencing data, and what does that look like? I mean, in the old days, it was chromatograms, and I kind of still wish it was. I miss Sanger. I love Sanger sequencing. But we're talking about image files for Illumina, right? The actual base pair calling is coming from these completely impenetrable constellations. Or if you're doing long-read sequencing, right, it's those voltage timescales, right, about how the DNA is passing through those nanopores. Those aren't very photogenic, so I didn't include a picture, but they're big. Those files are big. But when we're talking about raw sequence data, if I'm writing an SOP, what I'm talking about is FASTQs, right? I mean, no one is, if you're doing nanopore, you are starting from the pod 5 sometimes. But for the most part, you're not starting from the raw images out of the sequencer. You're starting with a FASTQ. But I do want to take this opportunity, since I'm in front of people, to make my plug that base calling is not inconsequential. We shouldn't be breezing over it. It's a big deal, and it can really, really impact what's coming out, right? Especially as new technologies come on, like nanopore sequencing. I spend a lot of time stressing out about my nanopore genomes because I want to make sure those FASTQs are exactly what I should be getting out of them. So if we're just talking about data numbers, and I know this is boring and lame, but it's going to matter in a second. We're talking about 150 MB for the image files from a sequencing run like this. For FASTQs, a set of para and bacterial FASTQs, 50 MB. For bacteria, or sorry, COVID or other virus, 50 MB, maybe a couple hundred if we're talking about a bigger bacteria, right? If we're talking about nanopore, we're in the tens of gigabytes now, if you're including the pod 5s. And if we're talking about fungi, and you're sequencing something fun and exciting, It could be, 9, 10 times bigger than that, which isn't all that much data in the grand scheme of things, but it is a lot of data when you're restricted in where you're saving these sort of things. So we have to save this data, right? This is direct healthcare data. Some of it has very stringent retention requirements, 7 years for a lot of medical records. There are different times for different things. But also we have to have an analysis kind of perspective on this. If I'm making phylogenies of all of the group A strep in Rhode Island, I want it forever. I would like to have all of the data that we ever had. And I kind of plan on keeping it forever. And so that's a data set that just goes. There's no end. As long as people keep getting sick, it's going to keep growing. But what we do with that data can be limited by public policy. We can't put it everywhere. I can't just get more storage on my Google Drive and throw it in there. That's not how it works. It's got to go somewhere. We also have the analytical data, right? So we're generating all this raw data, and instead of just throwing it off to databases, we do stuff with it, thankfully. And what stuff are we doing with it? Well, fundamentally, quality control. My job is like 90% as a QC person. I should hang out in the QC meetings more because, with genomic stuff, garbage in, garbage out is the name of the game all the time. And there's so many gains to be made with the data that's coming in. But also because I work really closely with the lab. And I was talking about this a little bit before. I give a lot of direct feedback to the sequencing core that I'm part of. I have all these little scripts written to like map sequencing runs back to the 96-well plates they were prepped on, or if it was multi-plates, to prep them back. I have an insane script on the one piece of automation we have, which is an OpenTrons that runs on like raw, straight Python. It's a great research machine, but it's not a great production machine because it gets a little messy. And so I have tools to map our stuff back out to the steps it took on that machine to try and look for these little teeny tiny places of improvement we could have in the laboratory, because I'm hanging out with the folks in the laboratory. And this is a great iterative relationship we can have to always get better. Our yields get better, our throughput gets better, the data gets better all the time. So even if we weren't submitting the data anywhere, I'd be busy as anything just working with the folks in the lab to make that better. It's great. I have a really, really great time that way, and it's something that I like to talk about probably more than I should in a setting like that. We still are generating several thousand SARS genomes a year. These are amplicon sequences. We do consensus mapping on that. There's a little bit of QC work to be done there, although a lot of the bugs for that have been worked out, but it wasn't always that way. We were kind of in a flat panic during the pandemic trying to scale this stuff up. But we get to do all these fun and exciting collaborative studies where we provide data sets to maybe some people at Brown who are doing some cool things with it. We're trying to get better at that. This is not something that we have been doing for a very long time. We haven't had the data to share, but we're trying to be sort of that partner where we can generate this raw data with a lot of rich metadata and then get it out to the folks who want to look at it. We're picking that up in wastewater and RSV and mpox and really as many viruses as we want. This is a really ugly, terrible genome that I had, which actually half of it's a two-step. I don't know if any of you guys have ever done any bench work with this, but the PCR will be in 2 different batches. Turns out one of them failed. We had a bad batch and we were only getting not quite half the genome because it was weird. But that was a real nightmare to figure out until I just mapped it, and then it was quite apparent. That's what a good genome is supposed to look like. And we can use it to actually find some really exciting things. This is actually an mpox genome, monkeypox genome. And there's a big gap here in the sequencing. And this is because one of our primers failed out, the primer set that we were all using. It happened here first for whatever reason, or maybe I just was loud first. But the primer set we were all using had a dropout from a common mutation, and it ruined the primer. And so we just weren't getting coverage there. When we had kind of higher automation through this, we weren't really noticing. It was still passing QC because it was only a very small chunk of the genome, but it was a chunk of the genome. We wanted to have that back. Sometimes you find things that aren't mechanical problems. This was an actual deletion in the genome. I was very excited about that one. I spent a lot of time talking back and forth about that. I have a pet obsession with deletions because they're not actually used in phylogenetic analyses, right? You can't compare what's not there, but sometimes what's not there is very informative. There are whole clades of things that you could draw a line on because there's something not there. There was a lot of cool stuff about that in COVID that I didn't get to investigate as much as I wanted to, but it's really, really neat. And if anyone wants to talk about it, I want to talk about it. Flu stuff. We do a lot of flu. I don't know if anyone's ever worked on flu before. I had not, and now I work on flu a lot. And boy, what a nightmare that bug is. It's so much fun. It's a segmented genome. We use these universal primers to get the 8 segments for the genome, but those segments have incredibly disparate histories. It's really, really exciting. But we don't use mapping to create those genomes. It's a cool iterative assembly program that the CDC gave us. It's a lot of fun. I'll talk about that later because it'll come in from cool research stuff that I'm doing. But just calling clades and stuff like that. That's what we do with that. As far as actual volume goes, like not number of sequences, but number of work and amount of data generated, it's all in the bacteria, right? We're expanding this program all the time. We're always adding new systems to be working on. Antibiotic resistance has become a huge thing because the databases that we have, AMR Finder and CARD and all these things, are fantastic, and they get better every single day. And they might not be a replacement for real-world antibiotic sensitivity testing, which we do all the time in the lab, but as a backup or as like a red flag for, hey, you should take a second look at this thing. It went from not doing it at all to I get 2 or 3 of these requests a week now, just constantly going. And it's really, really fun and really informative. And the more you do it, the better it gets, the better the data set becomes. It's been really, really exciting. Relatedness. If I had an extra buck for every email I've gotten from our epidemiologists up the hill of just, hey, are these related? I could have retired early. And it's my favorite email to get because I am a phylogenetics guy and I love that question. And again, this is a question that was really hard at the beginning when we only had a handful of genomes. Are they related? I mean, I don't know. These are the 2 closest gonorrheas we've had yet. But when the data set is 10, what does that mean? But when the data set is 500 and I've got time data and location data and there's, partner or transmission tracking and things like that going on, suddenly this becomes a really useful phylogenetic data set. And then when it becomes a really good data set, you can go back out to your partners, to the hospitals and to the doctors who are working on it. And we can say, this is what we can do now. Imagine if you could get us, every sample that comes through the door. And the data sets just sort of balloon after that, and we get really, really cool things. I have HAI down there because I've been doing a lot in hospital-acquired infections. And as far as like direct applicability goes, because we're doing this sort of surveillance thing, hospitals care a lot about their infection control programs. They care a lot about people not getting more sick when they're in a hospital. And it turns out that that is a spot where public health genomics can really shine, because we can take a lot of sick people with a lot of disparate sequences on there and tell you exactly who's related to what. And we've had some really, really, really cool outcomes from that, but I'll talk about that before. And then unknowns, those are less common and they don't happen that much, but it is kind of fun when you get a panicked email. It's like, we don't know what this is. And it's like, I can tell you. And a quick sequence and run against the Kraken database or something like that, and suddenly we've got an answer. It's a lot of fun. So I said I was going to talk about challenges, and I've already alluded to this, but challenge number 1 is storage. Reports aren't that big, but intermediate files are. My analyses tend to balloon, and they can really, really balloon. And if I'm only putting in tens of gigabytes of input, sometimes I need hundreds of gigabytes of space to do the analysis, which doesn't sound like a big deal, except the primary drive on my analysis computer is 1 terabyte. So I better not be storing anything else on there. And we have a question all the time of what must be saved. And what must be saved and what should we save are 2 different things, right? Because we want to have these recurring analyses. But we also have something that I thought about a lot before I came into a medical setting, but I hadn't really thought about that much, which was reproducibility. When I was doing thesis work, reproducibility for me was I got to make sure I can get the same tree every time, right? If the data came back and I ran this again, I should be getting the same relationship. And that's more of a consideration of like, have I considered all the variables? Is the science good? Here, that's not what I'm talking about. It needs to be molecule for molecule exactly the same. I need to know what versions of the software I was running. I need to know what was in the database, in the reference database. I need to know what the data set I was running at the time was. I need to be able to go back 7 years and give you exactly the same analysis that I gave that day. And I need to have all the data that I need to know to be able to recreate that. Because I'm not freezing everything in Amber. I keep my software as up to date as I can, which for research software sometimes isn't that much because it hasn't been supported since the postdoc finished 10 years ago and it's been untouched. But sometimes it changes a lot. And so it's a whole new series of considerations that I had not thought about a lot before. So having this documentation around your results of exactly how you ran it becomes super, super critical. And then also making sure that you can go back and run it that way becomes very exciting. So I promised I would talk about some fun stuff, and I actually have time because I'm not going too slow this time. So we are doing some very cool things. Some very cool things that I'm really excited to talk about. Every time our data sets get bigger, every time we add more data to this, we can do more stuff with them. And I get to show up to every meeting that I go to and be like, hey guys, the future is unlimited. This is an optimistic talk. We're growing as fast as we can, and we want to. We're so hungry to be applying more data and getting more information for this. And so my phylogenies are getting better with more background data. And I'm now at the point where I'm saying, look, we've gotten as far as we can get with the clinical data. Please give me environmental data. And before, the buffer to that was, well, this isn't really something we routinely collect. How is this going to come into play? Is it going to be useful? Now that I have the robust data set and I can say, look, we identified this, we did this, it could be this much better with routine sampling of whatever. the case is being made, and we can change the legislation, or we can change the guidance, and the data starts coming in. It's really exciting. And every time we do that, we can do more complicated analyses, and they get more fun. they get better to talk about. But they come with their own challenges too, which is challenge number 2, which is infrastructure, which I have talked to everyone who would talk to me about that today about. And that is because this is our computer infrastructure. It is my desktop workstation, which is also all our storage. We're talking one Linux workstation. I'm trying to make it 2 so I can have a production in a dev environment, but I don't right now. It takes a lot of time for me to take tools and workflows and methods that were written for an HPC and make them work on this. It doesn't mean that you can't do it, and sometimes it's plug and play, but it isn't always. And that is time and effort that I'd kind of like to be spending doing the analysis instead of thinking about that. Although I'm lying a little bit about that. I kind of love making big data things work on a tiny computer. It's a weird hobby I have. I was a Linux nerd before I was even in college, so it's like a chronic problem for me. But it's really exciting. And you spend a lot of time doing these interesting workarounds. You got to get weird with parallelization. You got to chop things up into smaller pieces. You can't do whole genomes at a time. You got to get creative ways to pare down the analysis and then find ways to check those pared down analyses against bigger ones and make sure that you're getting the same answer, that you can confidently do that. And believe it or not, I told you it was going to be important later. This is where my mycology degree comes in. Not because Clark, where I did my PhD, didn't have the resources. Actually, we did have a computing cluster, and it was very well supported, and there was some really great people there doing that. It's because I was working on dikaryotic fungi. I was working on the genus Lentinula, which is what shiitake is in. It has 2 independent genomes in every cell doing their own thing, expressing their own stuff. They're each 46 megabases. It's big, it's unwieldy, it's full of all sorts of horrible recombination. And software is not always written with fungi like that in mind. Some things have like chromosome thresholds that are too low to use with that. Some things have size thresholds. There's all this stuff that you would never think about just running this, I mean, I say off the shelf, but out of GitHub software, that might not apply to it. And you have to spend a lot of time thinking about that and trying to make sure that it's going to work. So I had a lot of practice doing this, and I actually love it, like on its own, doing its own thing. I think it's a wonderful exercise. And so I get to do that every day. I'm as happy as you can possibly be trying to make things work on my computer. Not that I'm happy with it staying as the computer. I'm selling the computer a bit low. It's an OK computer. I got 20 cores and 128 gigs of RAM. You can do some stuff with that. It's not a computing cluster, but that doesn't mean we can't do exciting things with it. For me, and a couple people have asked me this question, so I feel like I should clarify. When I'm talking about software, most of it is shared software, Nextflow software from other labs or from other institutions. Like you get this stuff. It's not off-the-shelf commercial software. I don't even know if that exists. A lot of it is really horrific bash scripts that I really hope that none of you see, because boy, are they just cobbled together of nonsense with tools from all over the internet doing good knows what. But sometimes they work well and they stick around. My mpox pipeline was something that I threw together kind of as fast as I could because we had the data and we needed to analyze it. And after a little bit of tweaking, I'm actually really satisfied with it, and I haven't changed it because it worked great. That's exactly what it's supposed to do. Same thing for a lot of these things. They are. It's a lot of fun. It's again in its own challenge. And for the hawk-eyed of you, which you actually can't see because of the resolution, if you look up there, it's my favorite book, the awk programming language. I use it all the time. It's so much fun. You know, finding creative ways to get things done. It makes you think about it in a way that you can't just call up pandas and do something in Python easily, right? It's a good exercise. So I mentioned Nextflow. I think overwhelmingly the future, I'm trying to be good and learn Nextflow properly and actually containerize my code and do things, in a real way so I can share it with my peers the way they share with me. But this has been a game changer. I haven't been doing this for that long, but I have been doing it for long enough that I remember before we had tools like this, and the portability is incredible. If Utah writes a program, which Utah writes some really incredible programs, you guys should go visit their GitHub at the Utah Public Health Laboratory. If they write an Xflow library, it will just work on my computer. And maybe it won't be as happy as on their cluster, but it's going to do the same thing there no matter what, and I'm going to get the same results. And if the CDC writes a pipeline that way, it can do the same thing. I can string them together freely. They all talk to each other. Oh, it's magic. And I was saying before, the public health bioinformaticians, we're all in a big group chat. We talk all the time. We share software freely. It goes all over the place. We're writing this for the public, and talking about being able to share stuff with the public, I mean, I don't know. I can't talk enough about it. I need to learn more. I wish I had more time to sit around and learn so that I could actually get my horrible bash scripts into Nextflow, but one day it'll happen. This is one of my favorite Nextflow pipelines. This is all the software in it. This is all research software. If anybody knows anybody who wrote some of this, thank you very much. Some of it is very well supported. Some of it is very ancient. Some of it is very, very ancient. It hasn't been touched in about 15 years, but it all does important things. So just for an example of what we're all cramming into one single pipeline there, that's what we're doing. So finally, what are we doing with it? I have left enough time to talk about the cool stuff, and I hope that we get to talk about this afterwards. So every single pathogen that I have a data set for, we are trying to really push the analytical boundaries of what we can do with it. I'm very dissatisfied with just putting it into NCBI and hoping for the best. I want other people to do the work. What I really want is that we started the work and somebody calls me and says, hey, we're doing this really big analysis of whatever in Vibrio. Have you guys thought about it? And I can say, oh my God, you want me to send you my tree? We can put them together and see how they look. This is my dream. This is what I want. And so we're doing that in everything. So this is group A strep. Group A strep is probably the fastest scaling we've done since COVID. We were not sequencing group A strep at all. We were sending it to Wadsworth, who is our partner in New York. They're the regional center of excellence. They are very good at what they do, and they do a lot of stuff in-house. They are excellent scientists, but they do stuff when you can't. And now we can. So we are. So we had this group A strep outbreak, and our epidemiologists had really important questions like, is this outbreak actually an outbreak, or is it just that we're seeing a bunch of people with group A strep at the same time? Is this the same group A strep we were seeing before COVID? Could this be from out of state, or is this a local thing? Good questions. We didn't have answers to any of it, but we could. And so we asked the question, well, how much can you give us? And it turns out the answer was, when this analysis was done, 350 group A streps that were spanning about 5 years. The answer now, I think we're at 420 now, going back to 2015, I think, is our oldest one. Because you can save the bacterial cultures and then sequence them whenever you want. And thank goodness somebody saved them, because whenever we want is now, and we're sequencing them. And now that we are doing this great work with it, we're expanding the sampling. Now, every time we're getting a positive strep, or even during these routine screenings, we're like, no, save it, save it, save it, send it to the State Health Lab. Let me build my data set. And the data set is letting me do this. So this is a very ugly unrooted tree. Actually, I think it's a beautiful unrooted tree. If anyone's worked in group A strep before, you'll tell me, oh, there's already M types for us to differentiate these lineages. And there are. And I just wanted to see how those M types looked in the state. And so I did. When you just graph them like that, it's not a big deal. But like I said, we have all this metadata. We have all the patient information. So we get to do more stuff with it. So I can add as many layers of analysis over it as I want to. I can add the year that it was collected. I can add who it was collected from. I can add all sorts of really fun things that I'm not allowed to put on public presentations. But if you're working on the project, we could talk about it. And I can just visualize them and see how they're going. So the orange is a time scale from, this is the most recent, these are the oldest. And I was looking at clade expansions to see which of these groups is representing most of them. And then I started building a pipeline to do this routinely. So when I get a new group A strep, I got one yesterday, I only reanalyze that genome. When I get a good consensus, or rather when I get a good assembly out of that, that goes in the pipeline for it to be included into the big data set, stick it in the new big phylogeny, and then see if anything cool is appearing. And cool things do. Of all my data sets, this is where the most exciting stuff has come. We keep identifying these clusters where before it was one or two disparate things, and then suddenly two people come in and they clade out exactly the same, and they are 0 SNPs apart from a core genome that I spend probably more time than I should maintaining and making sure it's exactly how I want it to be. Because as I explained many times, we're hanging out with our peers, right? Core genomes influence the outcome a lot. And if you're a little too narrow on that, you're going to artificially inflate how related things are. So it changes with the population that we get. But it's a fun thing to do to stay on top of that. But when you get something that tight, 0 SNPs, 2 SNPs apart, well, that's a clonal transmission. We're talking about, that's one bug. And when you give that data back and let the epidemiologists do what they do best and trace that stuff down, we found really, really exciting things, tracing things back to a single piece of equipment or a single piece of personnel or a single location, like real-time things. And I can do this turnover now. I can have its placement in the phylogeny, by the next morning when we get, depending on what we're doing the sequencing runs. So we have this real-time feedback, and every single one of those contributes to the data set, and the data set gets better and better. And now we're tracking plasmids in the whole data set and where they're moving around. We're tracking weird genetic elements that we weren't even thinking about before that are coming through in these giant populations that we wouldn't have thought of. It's super, super fun. And this was 1 year. If I had given this talk a year ago, I would have had no group A strep to show you. And I have 400 and something now. It was really, really exciting. And I'm getting a lot more. I think we're on track to have 200 or 300 just new ones this year. So it's going to be really, really great. And then I promised that I was going to talk about weird phylogenetic stuff. And the timing is working out perfectly that I will end this way so we can complain about it more at the end. So this is flu. This is avian influenza. These are just the SRR accession numbers. You can look them up if you want to. They're in the public database. I got rid of all the fun stuff, unfortunately. I think flu is really exciting. I've been ramping up flu sequencing with my supervisor. Similar to monkeypox, we saw it coming through. We get a lot of testing, and we were like, well, can we sequence the residuals? If these are going through molecular tests, this is a treasure trove of biological data. Can we just start making genomes out of it? Yeah, sure. And we got the CDC protocol and started cranking out genomes as fast as we could get them. And I'm doing some work with our CDC partners for getting it into their databases. But while I had it, I was like, I would like to do something with this. And some of the questions we could ask are fun phylogenetic ones to use for a data set that's this big. So I was talking earlier. So flu is a segmented organism, right? It's got the 8 segments, and those segments have extremely disparate histories. When you talk to people who know how flu works, who have been in flu for a long time, they understand the different phylogenies of those component parts, right? So we talk about the H type and the N type all the time, but you could have a flu lineage, not an organism, right, but a virion, where 7 of its 8 segments have been together co-evolving for generations, 50 years or something like that. We have a new introduction of one of those segments, right? A new H type comes in from somewhere or something. Oversimplifying, but one of these segments comes in. Well, those 2 phylogenies are like fundamentally incongruent, right? They are different. And that is correct. When we think about phylogenies, we're thinking about an organism phylogeny, right? Does it have the one evolutionary history? Well, no, it doesn't. It has multiple evolutionary histories. How do we think about that? Well, if it's your bug and you've been thinking about this your whole career, you just have a bunch of trees. The flu folks have a bunch of trees. And when you ask them about the lineage of whatever infected this chicken that you've got now, they'll send you an H tree and an N tree and some analyses from the rest of the genome. But for everybody else, that's not for me at least, that's not an intuitive way to think about a phylogeny. I don't like to have that. But there are ways to think about that differently. So this is it's not astral, it's aster now, and this is actually waster, but this is a coalescent species tree. This uses a magic thing called quartet decomposition. So what I did was I broke the 4,000 flu genomes that I had, avian flu genomes, down into their 8 components, did alignments of each of those subsegments, picked the best evolutionary model I could for each of those subsegments, produced the best phylogenetic tree I could for each of them, a maximum likelihood tree, and then took those trees, decomposed them into their quartets, which is every possible relationship of 4 tips, right? It breaks it down into these weird little thingamajigs. It bins them, it piles them up, and it makes a summary tree, a tree of trees that displays not necessarily the phylogeny as in the actual evolutionary history, but the organisms that have the most similar evolutionary histories given the incongruence of the things that formed them, which is a long sentence, and I'm sorry to rattle that off. But it's a weird way to think about it. But what it gives you is a lovely unrooted tree. I had to take all my fun stuff off of this. But let me tell you, when there is some really cool geographic signal coming through on this, time signal, host signal, maybe one of these clades is cattle and another one is chickens and another one is wild birds, and it's really coming through in a way that you don't necessarily see in any of the independent segments. But when you put the whole picture together, it really comes out that way. It's really, really fun and exciting. I'm hoping to get something together on this. And I'm talking with a bunch of state vets, and maybe we'll publish on it one day. But if you ever want to work on stuff like that, it's my absolute favorite. If you want an even uglier figure, if you're like, you know what, no, this is too pretty and clean, you can always do splits decomposition, where all those competing incongruent histories result in their own connections. And it would take a real weirdo to want to sit down and trace that back. And I am that real weirdo. It's so much fun. So this is the same data set. It's saying the same thing. This isn't the 4,000, by the way. This is just a New England subset. There was too many. Doing a phylogeny of 4,000 genes, even though the flu genome is quite small on my machine, is about a week-long process. It's straight computation, and I can't do anything else on the machine while it's doing it. So I have to pick and choose when I do that, because sometimes I have to do stuff like group A strep and COVID. So I'm always trying to play with my infrastructure to do that. But that's what this is. This is the same data set. And these are splits decompositions, which are very similar, except they preserve those conflicting histories. And you get these parallel lines that represent connections, internal connections with different clades. If you've got weird genomes with lots of recombination or incomplete lineage sorting or something weird like that, these sort of kind of like esoteric phylogenetic things are just that they're so much fun to do. I don't know if they're going to tell you anything good. Sometimes you just get exactly what you would have gotten if you did like a PCA on the alignment, and it's not going to tell you much, but it's a really fun and exciting way to do that. And because we have all this data coming in, I kind of have the freedom to do these sort of things. So that is the end of my actual presentation, but I'm hoping that we can do a whole bunch of talking about the research, because we do a lot, and there's a lot of things that maybe I'm not going to be putting up on slides, and I don't want to present the data, but I do want to talk about the activities. And I want to be here to make connections with everybody and remind you that you have public health labs here. There's one in every state. Sometimes there's more than one. And we're all doing really cool stuff, and we want to work with you. We have gobs of data, and we don't have time to work on it. And some people could work on it for their job and then tell us how it worked out. It would be great. So yeah, anyway, thank you for letting me come here and ramble for 40 minutes. And please talk to me about your molecular biology and your sequencing.
Yeah, great talk. Thank you. It seems like what you presented, it seems like your work is somewhere in between a postdoc and a PI. Like, can you talk about, I guess, how you feel about this dynamic and maybe like what you're doing now, how that would compare with a postdoc versus like a traditional faculty position?
It's a good question. I don't spend a lot of time thinking about the lab in those terms. It's difficult. I had not really considered doing stuff outside of academia, except maybe like going back into plant pathology and driving a truck again for a long time until I was doing it. And so it kind of came as a surprise to me how differently things were organized. I guess the answer there is, it feels a lot like an early stage PI and that you're trying to build a program, but the program is entirely driven by what's happening around me. I'm not trying to reinvent the wheel. I'm coming in with a certain area of expertise. I would not consider myself an expert in computational biology by any means, but I have the time and I have a desk. And so I can do these analyses if you want me to. And my job since I've started there has been finding other programs that are already doing this work and saying, if you would like, I could try and do this. Would you like me to do that? And so it's almost like, as a service, we call ourselves the sequencing core. It's not quite officially our designation because we've kind of amalgamated off of the molecular biology lab. But we are operating in that sort of a capacity where it's other programs, other groups are saying, hey, you could apply sequencing to this. Can you? And then it's kind of this team effort to scale it up. I never did a postdoc, so I can't really tell you how close it comes to that. I avoided the whole postdoc world. I don't know. It's a lot of fun. As things are leveling off, I spent a lot of time getting the routine things working. I needed to have a pipeline for when a strep comes. I have them now. I'm spending more time thinking about publication, and we are encouraged to publish. More than encouraged. This is how we branch out and meet you guys and do things in the real world. We're starting to think about research questions. I don't know. It's been great. I do feel like I'm running my own little lab, but I'm one cog in a very tight, slightly larger lab, which is also one cog in a very tight, slightly larger lab. Like it scales up. Yeah, I don't know if that was a rambling answer, but I guess that's it. Yes.
I was wondering, with your perspective on some of the data challenges, if you had ever used or were permitted to use cloud computing as an option. I know in particular that Qiagen has been developing workflows for public health.
So,
and yeah, just your perspective on that.
So I said in the abstract that I was going to have a frank discussion about these sort of things, so I will. The state moves slowly. They have a lot of considerations for things. Everything has to be vetted. The powers that be go into these agreements with the understanding that when we make a decision, maybe it's not a permanent decision, but it's a very long-term decision. It's not this fast application. So we don't have any cloud at all right now. The closest thing we have to cloud is that our sequence data goes on base space before we take it off the Illumina sequencers. And even that, IT is not so thrilled about. They don't like it. They would love it if it came straight home. We've had a lot of conversations about this. We're moving towards the cloud. We have to. So my PC right now, it was kind of a joke that I had it up there, but genuinely, I got a terabyte NVMe that I do the analysis on if I can, but I can't do my nanopore analyses on it because they need more than a terabyte when they expand out, especially if I'm doing hybrid sequencing, or rather hybrid assembly. If I'm doing the long read and the short read at the same time, it gets too big, at least for the data sets that I'm trying to run. So I have to run them on my 8 terabyte solid state that I purchased, but took a very long time to purchase. We're not used to buying that sort of thing regularly. I have 2 1 terabyte spinning disk drives that I run as a RAID for backups. I've begged, borrowed, and stolen 7 terabytes on a different lab's shared network drive. And I have 3 or 4 external hard drives that I back up our real-time sequencers from, and I back my whole system up on some borrowed space from somebody else's shared network drive. But it doesn't live anywhere right now. And every week I'm in a meeting or two, not fighting for the cloud storage because they know that we need it and we're moving towards it, but reminding them of what the use case will be. Because this is a new program. 4 years, 3 years is no time in the scale of state operations. But also, this kind of came out of nowhere. Nobody asked for this. It kind of happened. I guess the feds asked for it. They really wanted us to be doing more molecular, but it has occurred. And so we're working on it. We can't use Theogen right now because that's on Google Bucket, and we don't use Google products at the state. So I can't use Terra. I don't know why we don't use Google products. They don't have it on anything else. Yeah, Peter's nodding his head. Somebody is concerned about the security. I know that there are some federal programs that use Google Bucket, so I don't know why it's good enough for them and not good enough for us, but somebody has seen something that alerted them there. They were working on an Azure implementation at one point, and I was hopeful that we would be able to use that one, but that has not come to fruition yet. There's talks right now about AWS. I would love to get some stuff on AWS, at least Glacier storage, to back up all my sequencing data, because my desktop is not where I want to have 8 terabytes of sequencing data. But we'll see how that moves along. Some places have no cloud access at all. Some states have laws against that. And I do have some peers that I talk to who have servers under their desk that they store all their data on. It's all local stuff. I was asked if I would be amenable to having a tape storage drive under my desk in the new building, which maybe. Yeah, probably. I will say a great deal of progress has been made this year, and I think that we will have stuff in the cloud very soon. And I will also say, even though I can't use them, the folks at Theogen have been so helpful. And their stuff is all just containers running on their platform too. And so if you need something that they have in Terra that you don't have, they'll send you the code, and you can do some stuff to make it think it's running on Terra and run it locally or on whatever compute you have, and it works great. And they've been lifesavers with that. So yeah.
The other just general comment is, and again, I don't know if it's an option, but we've approached this challenge by just pushing more data into NCBI as a backup.
Yes.
And also kind of sadly giving up on the raw data from nanopore, which they've made more and more difficult to push, like the past fives into NCBI.
Yeah.
So, yeah.
Yes. Everything goes in NCBI. Everything. Fundamentally, as a public health lab, we are providing data to the public. There's stuff that I have sent to NCBI that were part of programs or initiatives that were so data sensitive, and we are a very small state, right? We have to make considerations that other states don't have to, because if somebody has Legionella in New York, it could be one of however many. But if somebody has it in Rhode Island, it doesn't matter how anonymized I put that data up. If you know the year, you might know the person who had it. And we can't be outing people like that, right? So we have to really work to anonymize this data. But even if you have to send it to the feds who wait some time and upload it as part of another bulk thing to make sure you can't trace it back, it goes there. Every single genome goes to a public database so that somebody has access to it. But even so, we want to have a copy, because we can't, even if you ask NCBI, they will tell you, do not use us as your long-term backup, because that's not the role that it's there to serve, and we don't use it as that role. But also, there is a bias to that. It's only the stuff that passes QC that goes to NCBI, right? I say every genome, but I mean every genome that passes QC. And there's a lot of data that's just not good enough to go to NCBI. But we hold on to it for other reasons, because sometimes we go back and reassess it. We did that with COVID a few times, where there were genomes that were not passing muster because they had enormous amounts of private mutations or things like that. And in revisiting them when I had a minute, it turned out that that was actually a computational problem with software that we were, not my software, commercial software that we were using that was doing very bad things and inserting reference into high variability regions of the genome, which is a real no-no. And so after identifying that, I went back and I recalled those genomes and got usable genomes and resubmitted them later. But I wouldn't have been able to do that if they had just been the SRAs that we had submitted that way. Yeah, and there's not always a good place to put everything in NCBI. Sometimes you don't know where it goes. You know? Yes?
You have a question in the chat.
Oh, I do. Thank you for sharing the COVID-19 data. Oh, the QC process. All right, so the QC for your internal samples and external samples. So this is going to be a major cop-out. Every opportunity that I get, I outsource the QC that I'm doing to somebody else's program. I do it, but I don't want to make the decisions. And that's just because I don't have time to sit around and refine these things. Sometimes I do, and for some of our data, I have done that. But if I can manage, I find somebody else to pass that buck to. So for our flu data, we're an atypical lab. We're not a flu center of excellence. We're not submitting the data through these LIMS systems that the feds usually use for flu data contribution. We're sequencing it ourselves. I analyze it myself with my own version of the CDC's IRMA pipeline, which is this iterative assembly pipeline. And then they have their own tools for doing lineage calling and stuff like that. And then I submit it that way. But the step in between there, all the QC thresholds that we're using, all the output that we're checking, once we got up and running and sequencing, I sounded the alarm bells and I had a meeting with CDC. And I said, what are your thresholds? What are you using to call this from your tool? Please tell me. And I will stick to them as hard as I can. I do the same thing for all of our bacterial sequencing stuff. The CDC has a really great tool called Phoenix, which is supposed to be for HAI and some AMR bugs. It's supposed to be for these outbreaks associated with hospitals. It has all its own internal QC thresholds. It gives you a report whether or not it passed its thresholds. And we sat down and we said, do any of our other projects that I could possibly use this for, any of our other bacteria, need to be higher quality than what the CDC wants for its HAI? And the answer was no. And so I have deferred to that. And if it passes muster in Phoenix, then it's good enough for that. For COVID data and Mpox data, I do adapter trimming and I don't know. I have the whole list of the regular deduplication and trimming and removing weird ambiguous sequences and stuff like that. And then when it comes out, before I dive into it real hard, I throw it into Nextclade. And Nextclade's pipeline does have its own sort of internal QC thresholds. That's actually where it cares a lot about the private mutations and like long contiguous strings of ends and stuff like that. And I won't submit anything to GenBank or NCBI or GISAID. I do still send COVID to GISAID, don't I? I won't send anything to those if they don't pass the QC that Nextclade uses. I've kind of outsourced to that. I was doing a lot of internal thinking. I don't know, we have sort of internal running things about sequencing depth, what we'll consider accurate or coverage, but it depends on the bug. But frequently we'll outsource that to whatever product it's for. That was a very long answer for that, I'm sorry. Okay.
My general question is that you have so much cool stuff going on that I'm wondering how you prioritize. But I also have a specific question, which I guess from the purely computational side, have you, and with the storage question, have you explored other forms of compression? Because I know there's those algorithms out there like Spring and Repack or whatever that are supposed to
Yes.
use smaller files.
The answer is a little bit. Actually, I was at an APHL, the Association of Public Health Labs, had a meeting this past fall in Georgia that I was at. Actually, I was presenting on the Group A strep, and there was a group there. I think they were, I don't know if they were fellows or if they were. It wasn't like forever CDC staff, but it was some group in a computational branch who had done this huge comparison of compression algorithms. They had like 20 on all sorts of different sequencing data, who did what where. I keep privy to that, and I talk to a lot of folks about it, like in the State Health Lab sharing. We've done a little bit of it. Like I do everything with XZ compression that I can. For compression and decompression, actually, I run into some computational challenges with it. I use like pigs to do it in parallel quite a bit. But frequently, that's like the highest CPU usage of any step in my analysis, and it can take a big chunk of time to compress and decompress all these genomes. I think about it a lot. Yes, I've been thinking about it. The biggest challenge I have is that nanopore data doesn't compress very well right now, especially the fast queues don't compress well because their quality scores are all over the place. Illumina data, you'll have the same quality score for entire reads, and it compresses down by enormous percentages. I can't get more than like 6 or 8%. Yeah, and so to be honest, we are not doing a lot of nanopore sequencing we would like to do because I don't know where I'm going to put the data. And then asking about the prioritization. Personally, if you didn't pick it up from the rambling and arm waving and stuff, I have a hard time prioritizing what I'm going to do when. My supervisor is really good at that. And I am a much more effective scientist because I work for her. But also, genuinely, I do a lot of this stuff in response to questions, and a lot of them have turnover times. For certain bugs, certain E. coli's and salmonellas and listerias and stuff, we have to report them within a certain amount of time, which means uploading the data to a federal database. And I try to have my whole analysis done by then. So if I've got 7 days to report this listeria, I have 7 days to think about listeria, and then I got to move on. So it kind of keeps me from getting caught up in one thing or another. Not that it stops it altogether. Thanks for asking.
Oh, I was also going to ask about prioritization. Great talk.
It's getting to be more of a problem because we can do a lot more, and there's a lot more to work with now. For a little while there, it wasn't a big deal. I had like 10 E. coli's, and it was like, what am I going to do with that? But now I get to go looking, and it gets dangerous. I would love to have the time. I would love to have more friends to come and work with me and divvy things up. It would be great. Yeah.
So you mentioned that for some of the projects you have like PulseNet and things, there are very clear sort of requirements,
yes,
softwares, tools you have to use to kind of report. But then I imagine some of these, like the unknown investigations you talked about, or inferring relatedness.
Yes.
Those are a little bit more, we don't, at least to my knowledge, have very clear industry standards on how we do that. So I suppose my question is, how is that thought about in the public health labs in terms of how much standardization do you need before you're comfortable reporting these things out to a hospital or to the CDC, and how much leeway do you have to explore and kind of innovate through that process?
Yes, thank you. What a good question. I wish I had talked about this on my own. So yes, for things that are prescribed, like PulseNet, like Genome Tracker, those are hard and fast. There are hard thresholds. We do competency trainings every year. Nobody from the lab can touch those programs until they have been, you take a test, you send the results off, you get your report card back, you get a stamp, they give you a passcode to get in. It's extremely regulated because they have to be exactly the same every single time. If it is not like that, it is completely the opposite. And in fact, by design, there are many of these programs where our federal partners will not tell us what software they would like us to use. They won't even tell us what hardware to use for the sequencing because they don't want to be biased. These are not things that they're prescribing. They are just there to collect the data. And then we're kind of left to our own devices. If you're left to your own devices, there are two levels for this. There's the research work, which is not strictly regulated, and that can only inform surveillance work. I can send it up to the epidemiologist and say, keep your eyes peeled. This sort of thing is going on. It never gets to a physician. The patients can never know that the test happened or what the results of that were because it is not regulated. And for that, it's up to the discretion of the researchers. So most of the work that I do does fall under that. That's why I'm an environmental scientist, not a clinical scientist. And for that work, it's about remaining internally consistent and having a QC and a refinement process. I'm always making those tools better. I'm always trying to get data sets from peer labs and running them and trying to see if I can get the same results. And eventually it's coalescing on sort of one method with your peers. I used to have a whole bunch of things smushed together to try to do the AMR gene finding, but Utah's program is much better and a lot of people are using it. And if we're all giving feedback when things get weird or when Utah asks for it, suddenly we're kind of falling onto a standard that we can compare directly between them. The level above that is if it was going into patient information, if it was going back to the physicians, and that's regulated under CLIA. And when you do that, you have to lock it down. You have to write the protocol as it's written. You have to have certification data sets, controls that you get sent that kind of freeze in amber. And every time something changes, even if it's the version of gosh knows what that's going into that, any little sub-process, yes, anything, any minute change in it, you have to either not apply it and make sure that your software is not automatically updating and frozen in amber, or you have to reevaluate from scratch and get exactly the same answer every single time. And one of the challenges with that is, as I said, a lot of these things are subjective to data set, right? Especially all the phylogeny, all the relatedness stuff. You get a different answer depending on what population you're analyzing, because that core genome is different, the relationships are different. It sort of inherently changes with the change of the data set. You can't lock that down in that function. The stuff I'm doing with flu, the stuff that I'm doing with even some of the group A strep stuff, some of the further out group A strep, could never be a CLIA analysis. I could never lock that down, and I could never provide that to patients. So we spend a lot of time thinking about how we could make a CLIA program sort of as a subset of that program and what it would look like. And that's actually sort of why we use a lot of really old, ridiculous stuff. That's why nobody actually, well, people laugh at me with the awk, but not that much. Because once you lock something down, there needs to be a big change for you to reassess that. So a lot of times it's just pulling other people's software, and it's quite old. So the closest thing we have to CLIA programs, we've gotten some things from Wadsworth for Legionella. They have a really interesting tool called LegioCluster for clustering Legionella cases. That has been locked down. You can pull in a set data set, you can validate that in-house, and then use it like that forever. And that's kind of what we're doing with that. But there's a different line. You have to be really ready to defend your decisions when you lock something down. Every threshold and everything, yeah, you got to be ready. So that's a slow process. We're not really there yet with a lot of our software. We will be, but not yet. Yep.
So, Sean and I are colleagues at the department, and about 6 weeks to 2 months ago, a nuclear warhead hit Washington, D.C. and Atlanta, and there were substantial amounts of data sets being bailed off to hard drive, various quarters of our hard drive, etc. Would you just briefly describe for those gathered here some of the impacts and how you've adjusted to the various problems?
Sure. The public health labs are all, independent operations, kind of on purpose, right? We're all doing our own things. But the federal programs are the glue that link us all together. We are the dendrils on the end of the phylogenetic tree, and they're the core of that. And everything went through them before. And even if it was stuff that we were doing on our own, there was always a talk back, right? So the group A strep work, that investigation, we were all, we pioneered that in Rhode Island. We were doing that all ourselves. And still, every quarter or so, we were meeting with some groups from the feds who were interested in this sort of work, and bouncing the practices off them, talking about other data sets. How does this fit into the national scope? A lot of that has kind of gone away. We don't get to have those meetings anymore. And we're still trying to figure out what the future of these programs looks like. Some of that is just that we're not having conversations. Some of that is the teams that were maintaining the software aren't anymore. There's a lot of open bug requests on GitHub right now for some of the software that have not been closed or answered or addressed yet. And so there's a lot of kind of working out where we're going to do that. People are cloning their own versions of software. I have forked my own version of a few things to maintain, to get quick bug workarounds. We don't know yet, I guess is the answer. We will see. The databases are still up. NCBI is still going. The data is still going where it needs to be. PulseNet is still humming along at full force, and so is Genome Tracker.
Do those people used to be at CDC or used to be elsewhere?
NCBI stuff is all NIH. Genome Tracker is FDA, and PulseNet is CDC. So yes, all of them. I don't know of anybody who's specifically gone. I don't, we're still filling it out. I think any of us who rely on these federal databases and stuff feel the same way. We just sort of don't know yet. We're planning for the contingencies for if we don't know. Like I said, there's a lot of communication. We're very blessed in the labs that we do talk to our peers so much, and we're sort of resilient in that way. And I think we're going to stay resilient in that way. But also, man, it's going to take a long time to catch up our friends at the federal agencies when they come back around. There's a lot of stuff's going to have changed. So we'll see. Yeah. Thanks for mentioning it, Peter. No, it's kind of the elephant in the room with all this.
I think there's coffee thing somewhere. Is that at 4 o'clock? Oh, were we supposed to go somewhere else? Keep chatting.
We don't need you guys a long time, but if you want to stay around.
2 days ago, so well, if anyone wants to hang out without food,
Thank you so much.
we can chat with Sean.
Yes, please. I'd love to talk about whatever you want, including fungi.
Yeah, if anyone wants coffee, there's free coffee anyway. The wonders of CMV.
Sorry about that. I scheduled those like months in advance and I had forgotten.
It said at 4 o'clock, like somewhere to so I was like, I think I forgot how to leave the room or like what the deal was. But so anyway.
Can I grab the